So welcome to this DeBacco University video. We're looking at determining cannabis sex using PCR, using the PCR method, which is advantageous because you can tell whether a plant is male or female well before it actually flowers here. So we'll go over some of the methods and some of the background of this polymerase chain reaction method, or PCR. All right, let's get into the video here. So first off, we're looking at male and female flowers. So cannabis is mostly dioecious. What does that mean? Well, for fiber hemp varieties have been bred to be monoecious. And when we use these terms, what does that mean? Well, dioecious means that it's producing either a female plant or it's producing a male plant. Monoecious will produce male and female flowers on the same plant. So with these dioecious, we're having these distinct female and male plants. Separating out those plants, those male and females at early development stage, is useful due to the influence of gender at agriculturally significant traits. Meaning if we were looking at producing cannabinoids, we definitely want to be selecting for those female plants and discarding those males. So we're eliminating the presence of pollination, limit the presence of that pollen to reduce the chance that seeds are formed, which would greatly reduce the total cannabinoids produced. Now, when we're looking at this PCR method, what's going to be utilized is going to be DNA markers. Several experiments have been done to focus on developing reliable molecular marker for sex determination in cannabis plants. DNA markers for the detection of male genotypes is utilized. And when we talk about DNA markers, you might be familiar with running things on a gel with electrophoresis here. We also have the utilization of these different primers that will bind to specific sequences if they're present and replicate them so that we can determine whether that small snippet of a leaf is from a male plant or from a female plant, because those plants contain all those genetic markers at the very early age. So those male genes specifically, the exact markers used can be debated, but in general, the concept is the same for all. If the markers for the male gene or genes are found in the plant, will not produce the high cannabinoid sought flowers that many growers are growing for. Meaning if we determine it to be a male plant, a lot of pollen is going to be produced, but not a lot of cannabinoids. Now, what I've produced here, kind of a horse example, looking at determining males or females. Keep in mind the Mare is considered to be an adult female horse, and a Colt is a young male horse. So here we can see the genetic markers, we could see our samples, we could kind of make a correlation of what matches with what here. Again, may not always be a perfect match, but we could clearly see S1 sample 1 here certainly looks like one of the Colt or a young male horse. And the same is basically applied to cannabis. Now, why would you use PCR? What's the advantage here? Well, research in polymerase chain reaction, that's PCR, and it shows could continue in order to find more advanced DNA markers for sex determination within cannabis plants. This allows not only for quick detection time, but also very accurate results, both things growers are looking for. The plant sample required is very minimal, so it can be formed on plants early on in the seedling stage to reduce wasted care, space, and time on plants that will not produce desirable end products. The reason why it can work on plants that are so small is it's using the genetic level, and it's able to screen for it really only needs one piece of DNA. PCR would go through this multiple thermocycler, this cycle run, to amplify, and it amplifies things on an exponential form. So technically, having only one sequence of DNA, it would amplify that to be a very large number to show up on an electrophoresis gel to be able to use for detection. Now, can the cost be justified? So this is one of those constant questions when we're looking particularly at PCR. Can we justify the cost of PCR? So while this can be expensive for some growers, some can justify the cost. For larger grower operations, we'll often set this up in-house, meaning they'll conduct these PCR treatments within their own grow facility. As after the initial cost, the reoccurring costs are actually quite minimal. Once you purchase everything, reoccurring costs are quite minimal. For medium to small operations, individual samples typically will be sent out for testing for a fee from a testing lab. And the great question is, in the cost to benefit analysis, is the cost of the test going to be less than what it would take to grow that plant to produce flowers to be able to identify it? And that is really up to the grower to look at for their operation, for their particular kind of cost, their particular potential benefits, to make that decision that's the best for their operation.